This video will go over how we can use southern, northern, and western blots to examine information about mutations. So we have already discussed southern blots and northern blots. And just to review, southern blots are used to determine whether you have a particular sequence of DNA in your sample. And this involves cutting genomic DNA with restriction enzymes, which are just shown by the scissors here, separating the pieces by size, and then using a probe to determine whether you have a particular sequence present. Northern blots are used to detect whether you have a particular messenger RNA present in your sample, and this is asking whether you are transcribing a particular gene or not in that tissue. So to do this, you would take messenger RNA from your tissue of interest, you would separate it by size by gel electrophoresis, blot it onto a piece of membrane, and then use a probe to base pair with that particular mRNA that you're interested in to determine whether that particular messenger RNA was present in your sample. For protein, we can also do the same type of experiment. So protein, you would purify all of the protein from your particular tissue of interest and separate it by gel electrophoresis based on the size. Then you would blot it onto a membrane, and here is where things get a little bit different. So proteins cannot be detected by base pairing with a particular sequence. Instead, companies and research laboratories can create antibodies that specifically recognize a particular protein of interest. And so rather than using base pairing as the detection method, for a Western blot, we would use antibodies to determine whether that particular protein of interest was present in your sample. Now, the combination of these three tools, the southern blot, northern blot, and western blot, can actually be used to ask what is happening at different stages of the central dogma. So first, I'd like to discuss what type of information can be obtained from these types of blots. The first thing is presence or absence. So particularly with RNA and protein, it tells you whether that particular gene is transcribed or not transcribed for the northern blot, and whether that particular gene is translated into protein or not for the western blot. Another thing these blots are useful for is to look for a change in the relative size of a piece of DNA, RNA, or protein. So if this particular band shown here is the wild type version of the gene, you might have a situation where you have an individual who has a mutation in that particular gene such that maybe 2 or 3 of the exons are deleted. If that was the case, you would see that individual having a smaller band compared to the wild type on a southern blot. That deletion mutation would also cause the messenger RNA to be shorter than the wild type messenger RNA, and so that individual would have a smaller band, lower on a gel, of their messenger RNA compared to the wild type version of that gene. Same type of thing would be observed with a protein. And then finally, we can also use southern, northern, and western blots to determine the relative amount of a particular messenger RNA, protein, or DNA. Questions I'd like to quickly go over are listed here. So for example, if you have a band on a southern blot, that simply tells you that that gene or that DNA sequence is present in your genome. And most of the time, you will see a band on a southern blot. Just because you have a band on a southern blot does not automatically mean that you will have a band on a northern blot. So the northern blot is telling you, again, whether that gene is transcribed or not. What about the second question? If you have a band on a northern blot, will you also have a band on a western blot? Most of the time, when a cell bothers to transcribe a particular gene, that messenger RNA will also be translated into protein, but not all the time. So cells have multiple ways of regulating messenger RNAs. So occasionally, if a messenger RNA is present, it might actually be repressed in some way so that its translation is prevented. And in that case, you may see the messenger RNA for a particular gene, but not see the resulting protein. For the third question, if you have a band on a western blot, will you also have a band on a northern blot? The answer to this is yes, because the way that the information flows in the cell is from the DNA to the RNA to protein. And so you can't skip over that middle step of making messenger RNA and get protein. Next, I'd like to go over two examples. The first example is Huntington's disease, which we've talked about previously in the course. So individuals with Huntington's disease have one mutant allele, which has an expansion of CAG repeats in the protein coding region of the gene, and they're heterozygous then for a wild type allele as well. And so if you looked at the wild type Huntington's gene, you would see that that particular gene is a particular size in the genome. This would be the wild type. We're going to call that the plus allele. And if you compare the mutant allele with a wild type, you'll see that it is actually longer. And this is the mutant or the minus allele. This is larger because it has these extra repeats of CAG in the protein coding region of the gene. If you looked at a southern blot, an individual who was wild type would have one version of that gene, and let's just say it showed up as a band at that size. If you looked at an individual who had Huntington's disease, they would have one wild type allele, and that would be the same size band as is observed in non-affected individuals. And they would also have a mutant allele, which is actually larger than the wild type. And so they would have two bands. This is the wild type allele, this is the mutant allele. If you then looked at the RNA that resulted from the Huntington gene, in the wild type individual, you would see a particular size of RNA, and in the mutant allele, that messenger RNA would actually be a little bit longer due to this insertion of extra bases in the middle. If you looked at a northern blot, a non-affected individual would have one messenger RNA for the Huntington gene, and just say that that ran at about that position on a northern blot. You would then look at an individual with Huntington's disease. That individual would have a wild type size allele due to their wild type Huntington gene. They would also have a mutant allele that was a little bit larger. And so this messenger RNA is physically larger in size than the wild type. And so these individuals have two bands on the northern. The same thing is observed in the protein. So the wild type protein would be a particular size and the mutant would be larger. And on a Western blot, if you looked at an individual who was wild type, you would see a band at a particular size on that gel. And if you looked at an individual who had Huntington's disease, they would have a band at the same position as the wild type protein, and they would also have a slightly larger band due to the larger protein that resulted from that original insertion in the DNA sequence. The second example I'd like to discuss is a mutation in which there is a point mutation in the DNA that leads to a splicing error, and one of the exons is not included in the processed messenger RNA. This type of thing actually happens in one of the most common cystic fibrosis alleles that is found in the population. So if we looked at the DNA for the gene, I'm actually going to write out an exon structure for this particular example. The wild type gene may have 3 exons, as shown here, and the mutant would actually also have three exons in the DNA. However, there would be a point mutation, and I'm going to just highlight that with a little asterisk right there. So that would be the mutation. What you would see on a southern blot is in a wild type individual, if you had a band about that size, on the DNA, you wouldn't actually see a change in the size of the band of that mutant allele because it's just a point mutation, one base to a different base. And so an individual who was heterozygous, for example, would have only one band. Both the wild type and the mutant allele would be the same size as the wild type. And even if you had a homozygous mutant individual, that individual would also have a gene that was the same size because that mutation is simply a change from one base to a different base. It did not change the overall size. On the other hand, if you looked at the messenger RNA, what you would find is that the exons are included in that final messenger RNA, and that messenger RNA in the wild type would include those three exons, whereas the messenger RNA in the mutant would actually only include two of those exons because this middle exon is not being recognized by the splicing machinery, and so we are simply splicing this exon together with this exon. If you would look at a northern blot, an individual who was homozygous wild type would have a band at a particular size. An individual who was heterozygous would have one band at the wild type size and then one band at a smaller size due to the inclusion of fewer exons in that mutant messenger RNA. An individual who was homozygous mutant would have only that smaller band because they have two alleles of that smaller messenger RNA. If you look at the protein that would result from this messenger RNA, the wild type would be a particular size and the mutant would be smaller because of that smaller protein coding region. If you then looked at the western blot, a wild type individual might have a band right there. A mutant individual would have one band of the normal size and one band that is smaller due to that smaller protein that results from the mutant allele. And the homozygous mutant individual would then have only the smaller size band.